In this video we will discuss about regulation of complement activity. Good morning. Welcome to YouTube channel. Bioconcept, learn with concept. Regulation of complement activity is an essential process to protect body from excessive activity of activated complement cascade. This regulation can be done by the 5 mechanism. In first, complement activity can be stopped by dissociation of C1 components. The C1 inhibitor, also known as C1INH, is a serine protease inhibitors enzyme. This enzyme dissociates C1 component, 2, C1R2S2, and C1Q. This enzyme also inhibits C3b and MASP2 serine protease. In second, decay accelerating factors promote decay of C3 convertases. In classical pathway, C3 convertases, C4b2A, is dissociated into C4b and C2A by the enzyme of decay accelerating factor, also called DAF, CD55, and CR1, and C4 binding protein, C4BP. In the alternative pathway, DAF and CR1 function in a similar fashion, and are joined by factor H in separating C3 convertase into the C3b and BB. In the third, factor 1 degrades C3b and C4b into inactive fragments. C3b is dissociated into inactive fragments by factor 1, joined with its cofactor MCP, CR1 and factor H, and C4b is dissociated into C4c and C4d by factor 1 and its cofactor MCP, CR1, and C4BP. In fourth, lysis inhibition was done by protectin and vitronectin's protein. This are found in plasma membrane, and inhibit membrane attack complex to bind, and polymerization in host cell membrane. Anaphylatoxins, C3A and C5A, cleaved and inactivated by carboxypeptidase N, B, or R enzymes on its C-terminal arginine residue. This process is helpful to protect host cell from complement proteins.